Welcome to Nexus Medical Media. In this video, we are going to have a basic introduction on molecular biology. We'll be talking about nucleotides. Right, so what are nucleotides? What you need to remember first is the central dogma of biology. What does it say? You take your DNA, right, and you convert it to RNA, specifically messenger RNA, and then to protein. Right, so do you remember the processes? Changing DNA to RNA is by the process of transcription. And RNA to protein is translation. Right, but this is a story for another day. Today we'll be talking about nucleotides. Right, so one more time, what are nucleotides? These are the building blocks of nucleic acids. Nucleic acids. The nucleic acids are DNA and RNA. These are the ones we are talking about. Right, so first, if you look here, there is a basic structure of a nucleotide. Right, so a nucleotide is made up of how many things? 3 things. A nitrogenous base, which is here. Here you can see a nitrogenous base. You have a deoxyribose, right? Deoxyribose is in DNA, but in RNA it's just a ribose sugar, right? So you just need to remember that, because DNA is deoxyribonucleic acid and RNA is just ribonucleic acid, right? So in this case, I just showed you where you find the nitrogenous base. Next is this ribose sugar and a phosphate. Right, so this is the nucleotide, but there is something else that you get when you remove this phosphate. It's called a nucleoside. In a nucleoside, just look at that S on side, S for sugar only, and then T on tide is for that T on phosphate. Right, so that's the first thing. Then next you need to know the nitrogenous bases. How many do you have? Can you think of their names? Right, so the first one is adenine. Next, guanine, cytosine, thymine, not thiamine, thymine, and uracil. Right, so this is represented by an A, G, C, T, and the next one on uracil. I'd like to change the color. I'll show you why. Right, so here on uracil you have this U. Right, so why? Because uracil, you find the uracil only in RNA. This is found only in RNA, but in DNA, on each place you find thymine. Right, so thymine, you find it in DNA, and uracil, you find it in RNA. Next is how these nitrogenous bases pair in these nucleic acid, right? Because they form hydrogen bonds, right? So adenine make 2 hydrogen bonds with thymine, and guanosine 3 hydrogen bonds with cytosine. Right, so if you are talking about RNA, A will be bonding with uracil, but this is the case of RNA only, but this is in DNA. Right, so if you can look at these bonds, AT, this double bond, is weaker compared to this next G and C, guanosine and cytosine, which makes like the boiling point will be high to remove this bond, right? So next you need to know how you classify these nitrogenous bases, right? So they are classified into two groups: purines and pyrimidines. Purines are big, they have 2 rings, and pyrimidines are small, it's just 1 ring, right? So if you look here, here you can see the purine, the structure of purine, and the important amino acids that you use to make a purine: aspartate, glycine, and glutamate, right? And glutamate. These 3 are very important. We'll talk about this in a separate video, right? But if you look at the structure of a pyrimidine, you can see it's 1 ring, it's small, right? So here you use carboxyl phosphate and aspartate, right? And in the pyrimidine you find cytosine, uracil, and thymine. This uracil, just to remember, I said you find uracil in RNA. And thymine, where do you find thymine? You find it in DNA, right? So the next thing that I would like to talk about is a deamination reaction, right? So if you remove the amine group on cytosine, you get uracil. If you remove the amine group on adenine, you get hypoxanthine. And if you remove an amine group from guanine, you get xanthine 5-methylcytosine, right? So these are just important deamination reactions that you also need to remember. And last but not least is uracil. What's the difference between uracil and thymine, right? So thymine, this shows that you have a methyl group, right? So if you add a methyl group here, you get thymine, right? So uracil plus CH3, you get thymine, right? So in the next video we'll talk about the synthesis of pyrimidines and purine, right? We'll talk about something called the de novo pathway. So stay tuned and thank you for watching.